is Riley Merkel, and I am an undergraduate student in the Schmidt Lab at the University of Pennsylvania, and today I'm going to be presenting a project I'm currently working on investigating a paradoxical role of amygdalar GLP-1 signaling on cocaine-seeking behavior. Now, this is a work in progress, as a lot of the results of this project have been hindered due to the COVID-19 pandemic, but this is where I'm currently at with my project. So, preventing relapse and promoting long-term abstinence remain the greatest challenges for successfully treating cocaine use disorder. Unfortunately, there are currently no FDA-approved pharmacotherapies to reduce the rate of relapse, and further exploration of the neurobiological mechanisms underlying relapse is imperative. An emerging body of literature indicates that central glucagon-like peptide 1 receptors may be the answer to this. So they have been implicated in reducing the rewarding effects of drugs of abuse and attenuating drug-seeking behavior during abstinence. In contrast, however, our preliminary results show that acute activation of GLP-1Rs in the central nucleus of the amygdala augments the ability of a priming injection of cocaine to reinstate drug-seeking behavior during abstinence. These findings are obviously unexpected and suggest that central GLP-1 signaling has differential roles in motivated behaviors depending on the circuit studied, and thus opens a vast new area of research in addiction neuroscience. So the overarching goal of the study is to further examine the role of the central amygdala GLP-1R signaling in cocaine behaviors. So under the preliminary data section, you can see a project that we did examining systemic administration of the GLP-1R agonist extended-4, and the results indicate that it attenuates cocaine-seeking behavior during abstinence, as represented by the decreased active lever presses of extended-4 versus vehicle during reinstatement testing. Since our central amygdala results contrast this, we decided to verify our preliminary data by expanding our N and our doses used of extended-4. So we followed a basic cocaine self-administration paradigm, 21 days of self-admin followed by extinction, and then reinstatement testing. Reinstatement testing involved intracranial microinjections of extended-4 via bilateral cannulae of varying dosages, followed by an IP cocaine prime, and then a 2-hour reinstatement test session. We found that intracentral amygdala extended-4 microinjections potentiate cocaine-seeking behavior during absence, induced by re-exposure to cocaine. So you can see in the graph under results, there's an increase in active lever presses when there is extended-4 directly administering the central amygdala versus vehicle. So in order to determine if inhibition of this pathway is responsible or is sufficient to attenuate cocaine-seeking behavior, we devised this chemogenetic DREAD paradigm as described under experiment 2. So it involves a CAV2 CAV2 CRE-dependent virus into the central amygdala, and either a DREAD inhibitory virus or a control virus into the NTS. So GLP-1 is produced centrally by pre-proglucagon neurons located in the nucleus tractus solitarius, the NTS, of the hindbrain, and studies show that cocaine activates the NTS, including GLP-1 producing neurons that project to the central amygdala. So in combination with CNO, the DREAD activator, and cocaine, this will allow us to selectively inhibit these NTS to central amygdala projections and see what the effect is on cocaine-seeking behavior. So we hypothesize that this will result in a decreased cocaine-seeking behavior, decreased active lever presses compared to the results of experiment 1. Similarly, we hope to identify downstream targets of this GLP-1R expressing central amygdala neurons. So the bed nucleus of the stria terminalis is an area of particular interest to us. It is a brain region that received projections from the central amygdala and is similarly recruited in both anxiety and drug seeking. Thus, the BNST is a relevant nucleus to explore in relation to the proposed GLP-1 amygdala circuitry, and given the high incidence of GLP-1R expression in the central amygdala, it is possible that central amygdala to BNST projections are GLP-1 expressing and play a critical role in cocaine-seeking behavior during withdrawal. So in order to test this hypothesis, we intend to inject Florigold into the BNST and then slice at the level of the central amygdala and use FISH techniques to label GLP-1 mRNA. Co-localization of Florigold and GLP-1 transcripts would indicate co-expression of would indicate that the GLP-1 projections from central amygdala to the BNST do exist. So in conclusion, activation of GLP-1Rs in the central amygdala potentiates cocaine seeking during abstinence, and these unexpected findings are truly innovative and novel as they indicate that GLP-1 has differential roles depending on the circuit studied and opens a vast new area of research. And more importantly, we hypothesize that there exists one mechanism by which cocaine promotes both drug seeking and anxiety through increased GLP-1 signaling in the central amygdala. That is, that GLP-1 promotes a anxiety, stress-induced cocaine seeking behavior. So further exploration of these paradoxical findings is critical towards developing a therapeutic intervention that targets central GLP-1 circuits to reduce cocaine craving-induced relapse. That is perhaps maybe a drug that is an agonist at one receptor subtype and an antagonist at another receptor subtype may be the future of treating cocaine use disorder. So for future directions of the project, we plan to verify the role of GLP-1 in the NTS to the central amygdala pathway by selectively activating the projections via an excitatory dread and intracentral amygdala microinjection of ascendin 9. So we plan to do this because we recognize that experiment 2 is not selective to GLP-1 expressing neurons. However, we thought that inhibition and attenuating this pathway was more clinically relevant than exciting the pathway. So we plan to use this excitatory dread and extendin 9 to verify that these are GLP-1 expressing neurons that are having this effect. Furthermore, we plan to further investigate the central amygdala to BNST pathway. Perhaps inhibition of this pathway can attenuate the negative anxiogenic effects of cocaine that potentiate cocaine-seeking behavior. And lastly, we plan to explore other structures implicated in both anxiety and drug circuitry, such as the basolateral amygdala. And yeah, that's pretty much it. So I would like to acknowledge, I would like to thank my mentor, Dr. Heath Schmidt, as well as the rest of Schmidt Lab, who have provided me technical and intellectual support throughout my endeavors. And I would also like to thank the Biological Basis of Behavior Department at the University of Pennsylvania for awarding me the 2020 BBB Undergrad Summer Fellowship that helped me and will continue to help me fund this project.